a process we can isolate the DNA from the cell. So DNA once isolated, when we got the DNA piece, what we will do is we will apply a restriction endonuclease digestion over them. So we will break those DNA into fragments. So restriction endonuclease on DNA. Then when we got these different fragments, small and large fragments, we will apply electrophoresis technique. Electrophoresis technique. Gel electrophoresis. So in gel electrophoresis, what we will do, we will separate DNA fragments. So what will be the basis? Mass, that is size, and charge. Charge due to its basic nature of the molecule, DNA molecule, and its size. So in electrophoresis, we will separate those different fragments. So what we will do next is we will when electrophoresed, when separated fragments, those fragments will be blotting will be done over that. Blotting, that is southern blotting. And in it what we will do, transfer these fragments over to the synthetic membranes, and those will be nylon or nitrocellulose. So once transferred to those membranes, we will now hybridize. That means joined to a probe, that is VNTR probe, and this probe will be this VNTR probe will be having what we call radioactive nature. That is, we will use a probe here, VNTR probe, radioactive probes. So the radioactivity of this probe, attached probe, complementary probe, complementary to those blotted parts of DNA. So this complementary probe gets attached to the transferred DNA fragment, and this VNTR probe will be now autoradiograph. So this autoradiography will be used. Final step. So the first step was DNA isolation. The next was restriction endonuclease digestion. Third was electrophoresis technique, blotting then, and then hybridization, and lastly autoradiography. Finally we can get the radiograph where there are so many bands. So meanwhile we can use the PCR technique. polymerase chain reaction, so that the DNA sample of a single cell can be amplified to the level where we can effectively do the experiment, effectively conduct experiment. So from single cell DNA specimens of the tissues from the crime scene, for example. So these were the things about the methodology of DNA fingerprinting. So now we would highlight some of the, copy number, something will this high copy number, VNTR. Why we used VNTR? Because of the high copy number. So the frequencies are 0.1 to 20 kilobytes of this DNTR, and these repetitions, high copy number means the repetitions will be from 4 to 40 repetitions in VNTR. So this is about the VNTR, that is equivalent to what we started with, mini satellite, which we satellite that was probed or hybridized with VNTR probe. And so these are mostly you have different. Mostly these are the different sizes, different sizes of these fragments were there. Different sizes fragments. That is the repetition of those units, those fragments or bases, number of bases in one fragment, one segment, and repetition 1 unit. So those were the different sizes, but in monozygotic twins, so these will be what? Same repetitions. Monozygotic twins. You cannot within two individuals, sample of two monozygotic twin individuals, you cannot identify this on the basis of what? VNTR. So there you will use SNPs, single nucleotide polymorphisms. So through that technique we can get to know about the twins, differences within the twins, and those monozygotic twins, how those occurs after the blastocyst splitting in two. So same zygote becomes two after the blastocyst split. So they are the same in their those repetitions. So far we judge it through VNTR. So for then we use different the differences can be judged in the twins through SNPs or single nucleotide polymorphisms. So now how the PCR, one more thing is that PCR, why we used PCR is that it increased the sensitivity of the experiment. And when we did the DNA fingerprinting, that was used for forensic applications, that the science related with crime, the use of science in controlling or for in the cases of crimes, the scientific tools is in the form of forensic applications. We use this DNA finger printing. So we use DNA fingerprinting in forensic applications to detect the crime activity, criminal activities. So this detectivity is through DNA fingerprinting, and PCR increases the sensitivity here in the experiments. So the next applications of this DNA fingerprinting will be in determining the population diversity. Or genetic diversity. So this forensic application side, determining the population diversity, DNA fingerprinting is used. We also use PCR for increasing its sensitivity. So now lastly, I will turn to the diagram where we will come to know how exactly we did the identification in DNA fingerprinting so far forensic application is concerned, or for matching any of the samples. So here I would like to draw 3 samples. So 2 samples here. This first sample, this one box is the number of bases, either in case of single nucleotide polymorphism, the single base, or some 2 bases, for example GT, CG, or something like that. So that repetitions, or G repetitions, so those units are single box, and these are the repetitions, number of repetitions. So one more thing I didn't discuss is that how we differentiate between mini and micro satellites. So those are differentiated on the basis 3 bases. First is the richness of AT and GC proportionality. AT-GC richness decides the characterization of the micro and mini satellites. The second basis is the length of segment. So this length of segment. And third is number of repetitions. So these are the repetitions. For example, here it is 11 repetitions. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 and 11. And now the second one, the allele, second allele is here, which is having repetitions 9. 1, 2, 3, 4, 5, 6, 7, 8, 9 repetitions. So this one is from paternal origin, one is paternal origin and one is maternal origin, because mother and father are two individuals and repetitions differs on individual basis. So of course repetitions will be different. These are the identifying characteristics, these repetitions. So these are the VNTR1 or chromosome 7 or VNTR1. Now the repetitions are like this, and this is the sample 1, DNA from individual, DNA from individual A. And the next VNTR is having 8 units repeated 1, 2, 3, 4, 5, 6, 7, 8, and the second allele is 7, 1, 2, 3, 4, 5, 6, 7. So this is chromosome, for example, chromosome 2 or VNTR 2. And the next one is, for example, 5 repetitions 3, 4, 5, and the next allele it is 2. So these are from the sample 1. And the next sample is for example, the sample B is this one. Three units and 4 units and 8 units. 1, 2, 3, 4, 5, 6, 7, 8 and 7 units. 4, 5, 6, 7. So these were the third VNTR is VNTR 1 or chromosome 7, and this is chromosome 2 and the chromosome 16. This is chromosome 16. So this one is chromosome 16, that is 5 units and 10 units. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So this is chromosome 16 or VNTR 2 and VNTR 3. these were the two samples. This is from the sample B. DNA from the sample or individual B. So for example, these were colored. So these were colored as green. So these were colored green. And these are colored red. These are colored as red. And the third VNTR is colored as black. Now, as we can see here, that these were the two samples, and now I just remove this one sample. Only one sample I remove. But before that, we just try to remember that these were the sequences. These were 3, 4, and the VNTR 2 were the 8, 7, and the third VNTR was 5, 10. So 3, 4, 8, 7, and 5, 10. So we write here 3, 4, 8, 7, and 5, 10. So these were the things in 3 VNTRs for sample B sample. Now we draw the third sample, that is from crime scene. Sample C.